Glycolysis begins with the molecule of glucose, shown here. A phosphate is removed from a nearby ATP and attached to the glucose molecule, creating glucose 6-phosphate. Then, glucose 6-phosphate is isomerized into fructose 6-phosphate. An additional phosphate is removed from another ATP, producing fructose 1,6-biphosphate. With the addition of an attolase, fructose 1,6-biphosphate then transforms into either dihydroxyacetone phosphate or glyceraldehyde 3-phosphate. However, dihydroxyacetone phosphate will eventually transform into glyceraldehyde 3-phosphate with the help of an enzyme.
Then a hydrogen is added to NAD+. A phosphate is then removed from glyceraldehyde 3-phosphate and added to an ADP to create ATP. The 3-phosphoglycerate is then isomerized. It later loses a water molecule with the help of enolase. Phosphenol pyruvate then loses a phosphate to a nearby ATP to create ATP. Pyruvate
Pyruvate binds with the coenzyme and NAD+ form carbon dioxide and NADH. Now, this acetyl coenzyme uses another coenzyme to synthesize citrate. Then, a water molecule is lost to form cis-iconotate. Water is then again gained to form D-isocitrate. NAD+ and hydrogen enter to form NADH, which is removed to create isocitrate dehydrogenase. Then, carbon dioxide is removed to make alpha-ketoglutarate. Next, NAD+ and a coenzyme are added, therefore NADH and carbon dioxide are removed. This becomes ciconeal coenzyme. GDP and a phosphate enter to produce a coenzyme NGTP, forming succinate. Water is added to make malate. Then NAD+ is added to produce NADH and hydrogen, which produces oxaloacetate. Bicarbonate and ATP enter to produce ADP and a phosphate. First, in the electron transport chain, NADH carries electrons from food into the inner mitochondrial membrane, releasing NAD+ into the mitochondrial matrix and a hydrogen into the intermembrane space through the protein complex of electron carriers. Then, FADH2 is added and FAD is removed to release a second hydrogen into the intermembrane space. Next, 2 hydrogens and 1 oxygen enter to synthesize and release 1 water molecule into the mitochondrial matrix and 1 more hydrogen into the intermembrane space. Lastly, in chemiosmosis, ADP and a phosphate are added, which releases ATP into the mitochondrial matrix and allows hydrogen to pass from the intermembrane space into the mitochondrial matrix.